Welcome, Life Sciences, to another exciting life science lesson. Right, today what are we looking at? We're looking at the end of microorganisms. We've come to the end of a section where we've traveled on a nice long journey, and we're just going to recap a little bit of what we've looked at in the past couple of lessons, what microorganisms are, what effect they have, etc. So let's just go and recap a little bit of those diagrams coming up, the characteristics, the structure. So what are we looking at? Okay, let's go on the journey that we were on with microorganisms. We started off, right, with viruses. Now remember, a microorganism is something that is really, really small. And usually you need a microscope to see it. Now that's the concept that we were looking at here. So we started off looking at viruses, and for each of our microorganisms, we needed to look at what were the characteristics, what did they look like, how could we identify them, and for each of those we concentrated on their pathogenic qualities. And what did I mean by that? We concentrated on the diseases they produced. So really, really small organisms, and we looked at these really huge big diseases that could have quite an impact, right, on our country and other countries. So after viruses, we looked at the concept of what a bacteria is, the whole kingdom bacteria, what a bacterium. Again, we looked at structure, characteristics, the disease. Then we looked at the kingdom protist, right, the microorganism protists. Then the last microorganism that we looked at was fungi. So all in all, we looked at the 4 different kinds of microorganisms, and the last lesson that we looked at was how important are all these microorganisms in our environment. Yes, they do cause disease, they are pathogenic, but they are essential and we cannot live without it. Okay, so let's get those terms going, right? Just a little bit of a recap. Let's see what have we done so far, and then we're going to work on a few questions later. All right, guys, so the first one we looked at was viruses. And when we looked at viruses, each of the things that we looked at, I don't know, you might have done the whole concept of classification, but viruses, out of all of our microorganisms, they do not belong to a kingdom. And the reason why they are so special is this concept over here, is that we looked at, there's a lot of discussion about it, but viruses are said to be non-living. And when we look at the kingdoms, right, our kingdom, we classify all living characteristics. So the viruses don't fall into a kingdom basically because they don't have those living characteristics. Out of all of our microorganisms, they are the smallest. Guys, they are the smallest, and they unfortunately need a living host. Right, so a virus always needs to attack something. It always is going to produce disease. It's going to be bad for you if I have to use it in simple terms. Our other microorganisms have got good qualities and bad qualities. Our viruses, unfortunately, usually do cause disease. Now, when we looked at the structure, you don't have to know, right, the whole thing of the virus, but we looked at 3 things in particular. And the 3 things in particular was in the middle, there is this DNA over here. All viruses have either DNA or RNA. It's called nucleic acid, and it's DNA and RNA. All our other microorganisms only have DNA. Then they have a little protein coat around them to protect them, and then some of the viruses have a fat layer around them to protect them. So when we have a look at the virus, you simply have a structure around it, right, and you're going to have this DNA in the middle. So that is the basic structure. All right, so when it comes to viral diseases, for the diseases, remember, if you look at your notes, if you look at the textbook, if you look at the lessons, we actually discussed a variety of them. But technically speaking, you need to know one well. Maybe your school, all right, only concentrated on one. That is fine, all right? But you need to understand the concept of the viruses, what can cause them, right, and what is the management. And the diseases that we looked when it came to viruses, we looked at HIV/AIDS, right, which obviously quite a large portion in our country are HIV positive, so it has an impact on our health system. Then we looked at influenza, the flu, right? Maybe during winter months, we're going to see a lot more people might have the flu. With the COVID that we've had, maybe that's also linked to that. And then we looked at another one, rabies, right? Not as common as the rest, but as I said, you need to be able to do one of those diseases. And for each of them, you need to know two things. What causes it, right? We know it's a virus. What does it attack? Because viruses attack specific things. Then what does it do? What effect does it? Go through your notes, go through the lessons. Each time it says, these are the effects. Then how do I manage it? How do I stop the spread? How do I treat it? Right? What can I do? What can the government do? What are the implications? And then we can give you any kind of viral disease that you might not have heard of. But if you know how a virus works, you can then use, all right, the knowledge about a virus on those different questions. Right, our next microorganism was bacteria. They do belong to the kingdom, Monera, right, the oldest kingdom. So they are the oldest living organisms. And then when we look at our characteristics, what are we talking about? The structure and the characteristics. Bacteria are prokaryotic and they are unicellular. What does that mean? Right, prokaryotic cells do not have a nucleus. They have nuclei material in the body, but then they do not have any kind of organelles. Going back to that whole concept. Bacteria, here we go. Some are useful, some are pathogenic, some are very useful. We cannot do without it. Now this is the key. This is a characteristic, guys. Bacteria can be treated with antibiotics. Viruses, no. Right, so no. So antibiotics are a medicine, a drug, right, whereby we are going to be able to treat bacteria. So because it's living, and it's a simple concept, we can treat it, right? And the treatment is obviously going to be with antibiotics. Also, because they are living, they can reproduce by themselves. Bacteria don't need, right, a living host to reproduce. There's one bacteria, and what it does is it splits into two. And that's how the bacteria is going to divide. Nice and quickly and efficiently, and we make a copy of itself. Another characteristic about bacteria, right, are these diagrams here at the top. And very often, we give a bacteria a name according to its shape. So you'll see here, if they're rod-shaped, we often call it a bacillus. You'll see when we do TB, it's called Mycobacterium, or tuberculosis, and it's a bacillus. It's a rod-shaped one. Some of them are round. We say they are coccus. And then we have spirilli. They're spiral-shaped, so the name tells a lot about itself. And sometimes there's one, it's a little bit rare. It's on one of the STDs, and it's a comma, right? It's a comma, and it's called a Vibrio. But it's not one that we often discuss. Now, guys, if you have a look at this diagram, it's often called a typical bacterium, because that's what we use for a single cell, the bacterium. And you need to be able to label this structure. You need to be able to label it. Sometimes draw it, okay, not often. But you need to be able to recognize it. What are the structures and what are the characteristics of a bacteria cell? Our next group of microorganisms was the protists. And this was the funny group because this kind of like had things that were totally unrelated into it. So when we looked at bacteria, the only thing in that kingdom was bacteria. the Monera kingdom. Now, when we look at the protists, we saw that they had very differing characteristics. So much so that when we looked at it, we actually divided it into two groups. So first of all, right, when we looked at the characteristics, what did they all have in common? Because as I said, let's do like a mind map here. So we have, right, a few traits. These are the ones. They are eukaryotic. So for the first time, what do we see? We see a cell that has a definite nucleus, and we have all these organelles inside because it's got to do a lot more functions. So it's a eukaryotic cell. Even though we say a simple single-celled, right, it is a bit more advanced than our prokaryotic. All of the protists live in a moist environment. Generally, they live in an aquatic water. Some can even live in really moist soil. And here, again, they also reproduce asexually. So that is the characteristics that they all had. All right, guys, so now we've got to look at the diseases that bacteria caused. Now, as I said to you before, you are given a variety of diseases that you need to study, and you need to choose one. I'm not going to show favoritism, but I would suggest that maybe you look at the tuberculosis, the TB. It is a disease prevalent in our country, and it would be a lot more, all right, relevant for you to study. The other bacterial diseases that we looked at were anthrax, right, which we get in cattle. We looked at cholera, which we can get if we eat food or we drink water that's contaminated. And the last one we looked at, plants also can get, all right, a fungus, and we call that blight. Our next group that we're going to look at is the protists. Now, protists are a weird kingdom because there are a lot of different kinds of organisms that fit in here. Bacteria was quite simple, right? The Monera kingdom, if you're a bacteria here. In the protists, we have quite a variety of organisms that we actually divided into subcategories, but we're going to look at that just now. So when I look at protists, right, what do I have? I'm going to have, all right, over here, my group of protists. And all these characteristics over here that I've got all belong to this group. But then I'm going to split into two, and I'm going to see the variety there. So when I talk about a protist for the first time, we see a cell that is eukaryotic. What does that mean? All right, it's a cell that has a nucleus, and it also has all of the membrane-bound structures that the cells can have. Right, protists still live in aquatic medium. They can't survive where it's dry, so they usually live in water. And like the bacteria, they also can reproduce, all right, asexually. That means they don't need a male and we don't need a female. So what are these two divisions that we're talking about over here? Right, so what happens is when we look at the protists, we can divide them into plant-like protists and animal-like protists. So not plants yet and not animals yet, but they have certain characteristics that make them more plant-like or more animal-like. So if we have a look at the plants, right, the plant group we call algae. So that's the single name that we use for them. We say that the plant protists, they are algae. And the examples over here are, you'll see at the top, are diatoms, because they're nice and single ones, or really, really big ones are the seaweeds. Now, what can the algae do? Guys, algae can photosynthesize. They've got chlorophyll, they're usually green, and they can make their own food. Now, when we look at algae here, you will see these are unicellular, and these are multicellular. These are microscopic, and these are macroscopic. So here, scopic. So what we've got is really small and really big. So that's what I say to you. When it comes here, we haven't got this continuation of everything's the same thing. Again, these are able to reproduce asexually, and as I say to you, they can be really diverse, really small or really big. One single cell or many cells together. And those are usually, I said to you, single cells, diatoms, and the large ones are the seaweed, but we call them algae. The animal ones we call protozoans. That's what we call them. The animal-like protists, there's a group, right? They're called, we say here, they are protozoans. And we're going to divide them into 4 different groups. Now, what else do they have that is a little bit animalistic, if I use that word? Guys, they are heterotrophic. What does that mean? They have to eat. They cannot make their own food. Whereas the algae could, the animal part cannot make its food. Right, so it needs to eat. So basically what the protozoa, they're going to be little unicellular. Guys, these are really small. They're not like the plants. They are one single cell, and they're going to swim around looking for food. And these are the 4 examples that we're going to look at. They can be parasitic. This is the plasmodium. This is the important one because this is the malaria that we can look at. The amoeba ones, right, they go and they eat their food, they slink around it. The paramecium, right, this is little cilia, they've got little cilia to move, or the flagellate, they're going to move via their tail. But the parasite one, that is the one that we really needed to look at. Also in this group are 2 really important unicellular organisms, are plankton. They are found in the oceans. They're a really important part of the food chain. And again, you get the plant type and the animal type. The plant, phytoplankton, the animal, zooplankton, because that's where you put animals in the zoo. Guys, when it came to a disease, we only looked at one disease here, and that was malaria. And as I said to you, it plays a very important part in our society. We have usually in the northern province, we do have a mosquito problem, and mosquitoes carry, right, especially the female, carries that plasmodium, the parasite, not the mosquito, right? The mosquito has that protozoa inside that it gives to somebody, right, when they bite. Our last group of microorganisms are fungi. Now, they kind of like also a little bit off scale here because some of them we can see. They're actually not microscopic. When it comes to mushrooms, when it comes to mold when your vegetables or in your things in your fridge or your lunch tin might get all that green furry, we can see it. So it's actually here we have a little bit of a difference to the rest. Most of the times we can see it. There is a unicellular one, right, and that is yeast. And yeast, if we looked at the importance, played a very, very important role. Guys, they're also eukaryotes, right? As I said, we can see them. They cannot make their own food. Fungi have got various ways that they need to get food. They live off dead organic matter. Some of them are parasites. They live on you, right? But they cannot make their own food, and they have a variety of ways in which they are going to get that food. Now, very important, fungi, what do they like? Oh, nice, warm, moist, dark conditions. That is where they are going to, all right, flourish the most. When we looked at the structure, all right, of fungi, we looked specifically at bread mold, and basically what it does, it puts all of these strands into the bread, and it's looking to get the glucose. That's what it does. It gets the food out, and when it needs to reproduce, it produces this long stalk, right? At the top, it's got all these spores in. Can you see that? And when it needs to reproduce, it bursts open, and the spores fall on the bread, and new fungi are going to then, right, emerge. Then we looked at some diseases. Again, there was a few that we could look at. You really need to concentrate on one. And we noticed that these diseases weren't killer diseases like the other ones we looked at. They were more of an irritation, right? Fungi could be treated with, right, and not like antifungal cream, usually quite easy to obtain and then to cure. What we looked at thrush, which was a yeast infection, right? We looked at rust. We saw that plants also can get a fungus. Then we looked at ringworm, which is not a worm, but a fungus that grows on our skin. And then we looked at athlete's foot, when we don't dry in our toes so nicely and it starts to nibble on our skin. Okay, guys, that's all for now. We're going to take a quick break and we'll be right back. Welcome back, Life Sciences. As I said during a break, it's always necessary just to get the stand up, do a little bit of wiggle, get the oxygen going, get the blood flowing, right? Otherwise, we just get so bogged down and none of that information actually goes into our head. So what have we looked at? We just did a brief recap on all the microorganisms that we have looked at. And now we're going to look at a few questions that we could ask you on these different kind of sections. Right, so let's start off with our very first question. I always like to start off with a bit of an easier question. When it comes to the microorganisms, you'll see, guys, pictures and information you're going to need to read. Okay, we have 3 diagrams here, so let's go. It says study the diagrams. We've got 3, and I'm hoping just by looking at them, you already can identify which they are. And they say to you they are all microorganisms, and then we're obviously going to answer questions on that. I've kept the picture at the bottom here so that we can just refer to it. It says name the group of microorganisms. That is basically the ones that we looked at, the kingdoms. So it was a virus, bacteria, protist, or fungi. That's what we're looking at of B and C. So if we have a look at B, we have a look here. What do we notice? We can see this characteristic DNA in the middle, and it's got a layer around it. So a B is going to be a virus. And C, right, what do we look at C? We see it's got these, looks like little, right, threads, and they've got these big, right, dark balls on the top, and those balls are going to carry spores. And the only one that has spores over here that we can see, that was our fungi group. Nice, easy question to start off with. Now, studying question, provide labels for one. Number 1 is our bacterium, and that is a flagella or a flagellum. You can use either two. Right, and that is the tail. Try not to use tail because that's not the correct one. Number 2, as we said, this is a virus. So let's just put here bacteria. All right, and we said this is fungi. Right, what was number 2? Number 2, I'm going to put here a nucleic acid. Because remember we said a virus can have a nucleic acid, and that can either be DNA or RNA. So I would have accepted either of those answers. Number 3 is that black ball that I see to you on top. It's a really dark color. That's what we see. And it holds spores, so we call it a sporangium. Right, if we go back to all our distinguishing characteristics, remember what I said? You had to know what a simple, typical diagram of each of the microorganisms would be. Okay, now the next one, give the letter. Guys, not the word, not the name, give the letter. Please read your questions properly. It's even in capital letters to tell you, which is not considered to be living. So out of the 3, which one did we say was acellular? They only asked you for the letter. That would be a nice big A. Is eukaryotic. Now, if we have a look, this is non-living, so it's not a cell. Bacteria are prokaryotic, which only leaves us with number C. Fungi are eukaryotic. So that is by a process of elimination. Now again, name the shape of the bacterium in diagram A. Guys, so if, nice little clue here, so if you didn't know it was a bacteria, they actually gave you the answer. And remember, bacteria have shapes. This is a rod-like one, and we call it a bacillus, or a rod-shaped bacteria. But we do prefer if you were able, right, to give its scientific name. Okay, now the next questions that we're going to start off, very often with microorganisms, we're looking at case studies, looking at giving us data, etc., reading from graphs, right, reading from tables. It's sometimes nice and easy because the answer is there. You just need to be able to read carefully and look carefully. Okay, let's have a look at the question. Right, whooping cough. Now, if you have never heard of it before, that is okay, because remember, what does it tell you? It's a bacterial disease. Okay, so that's really important. It's telling you it's a bacterial disease that affects some infants. I actually had it when I was a child. I can remember I was off school for quite a while because I got it. An investigation was conducted to determine, so as soon as we see to determine, that's the aim of our experiment. The relationship between vaccinating infants against whooping cough and the cases of whooping cough reported. Okay, so basically what you're looking for here is you're looking at the concept vaccinations. All right, that's what we're looking at. Do vaccinations have an effect on getting the bacterial disease? Now, when we looked at, when we looked at the functions, remember we said a vaccination is there to help our body cope when we get a disease, so that we might not get it as in such a serious way than if we didn't have the vaccines. Okay, so now the data relating was collected a long time ago, right, between 1965 and 1996. That's fine. We just need to be able to read from a graph. Okay, what did they look at? The following data was collected. Now, guys, this is quite unusual because you got a graph that's got two sets of axes. Have a look here. You've got one on one side over here. I'm going to change the pen color quickly. On the side over here, the number of cases reported. So have a look here on this part of the graph, all right? Number of cases reported is over here. Then on the other side, now I'm going to change back to blue, the percentage of infants that were vaccinated, they're on that side. So you've actually got two y-axes. The graph below shows the data collected. So let's see, all right, we're going to come back to this graph quite often. Let's see what kind of questions they're going to ask. This is a very common question when it comes to microorganisms, because we can use microorganisms for experiments. So the concept of a dependent and independent variable, right, is going to be quite important. Now, the first one, dependent. A dependent variable is the results. And our results, all right, are always going to be on the y-axis. Now, why did I say this is quite interesting is that for the first time, you've actually got two dependent variables. Now, those dependent variables are the following. Okay, guys, so as I said, we have two. So if we look at number one, I'm going to put number one on the side here, and I'm not going to write it all out, but we're going to write the whole description. Right, let me do it, so otherwise, because I can't write on this thing. Number of, all right, let me go back, let me do that, of cases of whooping cough reported. All right, there we go. What's the next one? Number two. We're going to write that out just at the top there. Percentage, I'm going to just do it nice, I'm going to cheat. Percentage of infants vaccinated, all right, against, and I'm going to just put WC for whooping cough. Right, so we have two independent variables. We're looking for two results. How many of the infants were vaccinated and how many had the cases of whooping cough? What was my independent variable? My independent variable is on my x-axis. All right, remember, I lie, all right? And that's exactly what the x-axis is down. It's downward. And that would be the year. Right, we decided on which years we were going to collect this data. We had no choice. Remember, I decide. We have no control over the results. Okay, guys, now the next question is you need to have a look at both of the graphs. It says, state the conclusion based on the data shown. Now, remember, what were we looking at? Is there a pattern between vaccination and children getting sick? Now, if we have a look at our graph, what do we notice? When our vaccinations are down, right, if you have a look over here, if our vaccinations are down, what happens after that? Our cases increase. So what is our conclusion? What is it all about? Is there a relationship between vaccination and children getting sick? And it definitely is. The conclusion is the more children that were vaccinated, there we go, or you can say it the other way around, right? The least, or the number of cases of whooping cough, right, decreased. So we saw, what can we conclude? That vaccinations help prevent the whooping cough, right? So that's what I noticed. Looked at our trends. When one went down, when the cases of children went up, it's because the number of children were not being vaccinated. Okay, our next question, right, is again an experiment. Let's see what Liso did. Liso carried out an experiment to find out what factors, right, would help mold to grow on bread. She took 4 slices of white bread, remember, all the same bread, and put different substances, this is what I'm choosing, on each slice of bread as follows. So on slice 1, she left it dry. On slice 2, she put some tap water on. On slice 3, she put some lemon juice. And slice 4, she put some sugar water. Guys, I hope you notice she used the same amount. Controlled variable. Controlled variable. Okay, fixed so that you make sure that it's reliable. Okay, each slice of bread was put in a Ziploc bag and kept in a dark cupboard. All right, so remember, mold is fungus. What conditions do fungus grow best in? Warm and dark and moist. That's why she put it in the Ziploc bag to keep all the moisture in. Right? And she left it at a temperature of 25 degrees for 2 weeks. She observed the growth of mold on the 4 slices of bread and worked out the percentage area of the bread covered by mold. The results are shown in the graph below. Okay, so if we have a look here, a nice picture of the different slices of bread. Okay, state a hypothesis for this investigation. Now, guys, I teach my kids to start off a hypothesis, which is a prediction, with the following words. It is expected that. Right? Because that kind of puts them in the right frame of mind to make a prediction. Now, tell me, it is predicted that. Look at the information. The bread with sugar, water will have the most mold. look, I used my information, because what did it show me? It showed me the sugar water. So I'm being lucky here. I'm saying, yes, I can see the results. It is expected that the bread with sugar water, there's my one variable, will have the most mold. There we go, independent variable, and there is my dependent variable. Nice marks, 3 marks for my hypothesis if I was going to give you the marks. The independent variable, I, what did I choose? It's always on, right, the x-axis. And what did I choose? I chose what substances to put on the bread. That's what I chose. All right, what substances must I put on the bread? Explain why mold did not grow on the bread sprinkled with lemon juice. Now we're going to have a flashback to maybe when you did biochemistry. What does lemon juice do? It changes the pH, right? And what does the change in pH mean? It's going to change how enzymes work. And here you're going to see, explain why mold did not grow. The lemon juice interfered, I'm going to use the word, interfered, all right, with how the bread mold, its enzymes, this is a difficult question, all right, could break down the glucose in the bread. Right, so guys, if the bread mold couldn't break down, the enzyme couldn't break down, right, so what happened was the mold couldn't grow because it wasn't getting enough food. This is it's a difficult question. It's a higher order question. All right, the second last question was, why was dry bread included in the experiment? Guys, why? It's a control. That's why. We always got to make sure we have a control. So we wanted to see what would make it, so we took out, all right, no substances. So to make it see, all right, what it was. We always have to have a control. Take away something to make sure what is going to grow there. All right, identify and briefly describe the type of nutrition of Rhizopus. Guys, it's saprophytic. What does that mean? Right, it needs to grow, it needs the hyphae to penetrate, or to grow into the bread to get, right, to get the glucose in the bread. See, it's heterotrophic. Another word there you might have used, it is heterotrophic, right? It cannot make its own food, so it needs to be able, right, to get its own food. Okay, guys, that's all we have. Just for now, we're going to take a quick break, and we'll be right back. Welcome back, Life Sciences. Again, you know what a break is for. It's there to stretch the legs, stretch everything, get the blood flowing, right, so that you can concentrate. Sitting for too long is just not going to be, all right, healthy for you. Okay, guys, we were looking at questions on microorganisms, and I don't want to waste any more time. I'm going to go straight back into the last question that we're going to look at today. This is quite a intricate question, right? I think it's quite challenging, but if you read the information that's given to you, right, you should be able to come up with the answers. Okay, guys, so I'm going to show you a flow chart. I'm going to quickly go to it. It's going to look like that, right? And it's got 2 methods of how we're going to, right, how people can help, vaccinations and immunizations with their immunity. So I'm going to go back and I'm going to read it to you. The diagram below, right, on the next page, shows 2 methods, right? We've got 2 methods which are used to give humans protection against diseases. Right, very important now. Method A shows active immunity. Right, active immunity is when our immune system needs to kind of do its job. Right, so we put things in our body and we let our immune system, right, deal with it. It takes a bit longer, right, but it lasts a lot longer. Then we're going to look at method B, which is passive immunity. Right, so it's kind of like something that's we put in, it's going to do its job, but it's not going to last too long. Right, method A is used against polio. That is a virus. Okay? Method B is often used against tetanus, and tetanus is a bacteria. Okay, so we're looking at two different kinds of microorganisms. Now, if you look at this, the answers are on here, but you need to be able to understand the process. Okay, guys, method A is active immunity. And that means that we actually put some of the virus in our body to make our body respond with something that's called antibodies. Now, over here, when we get to method B, this is called passive immunity. And what's happening here is we're going to put it straight into, right, an animal actually makes the antibodies, and we put the antibodies straight into you. So we don't let your body do the work initially, right? We want to deal with the problem there and then. Okay, so name the substances that are produced by the body which destroy harmful viruses and bacteria. If we go back, you're going to see one word we're looking at, antibodies. Right, as I said over here, that's what we want to fight. So the word that we're looking for over there is antibodies. And you got that from the writing piece. Right, so there we're looking at the antibodies. Why does method A give long-lasting protection against polio? Guys, it's because our body, right, makes the antibodies and we keep them. We keep them in the memory. I'm going to use the memory. We don't destroy them, we don't get rid of them. So if, for example, when we need them, so when we get a vaccine, it's going to stay in our body for a long period of time. That's what we're doing, long-lasting protection. Our body makes the antibodies and we keep them. That's what happens over there. We keep them. We don't get rid of them. They're there in case we need it. Method B does not give long-lasting protection. Why? Now let's go back to over here. If we go back, guys, the most important is this one, all right? The animals, these points over there, the antibodies are made in the animal's body, all right? Then they take the antibodies and they inject them directly into you, right? So what's here? Why does it not? It's made in an animal's, okay, let me put here, made in an animal's body, not in yours, and it lasts just long enough to help with the infection. Right, because what happens after it is, right, our body breaks it down or destroys it. We don't keep a copy. Right, we don't keep a copy for as long. The tetanus one sometimes can last us for up to 5 years, but the polio will usually last us a whole lifetime. In immunization against polio, a second dose of the weakened virus given. This is called a booster. Guys, why is this booster necessary? Why do you think? It would increase, right, our antibodies, and it would give us, and I'm going to use the word boost, give our bodies a boost. Right, what does a boost mean? A boost means extra protection. That's what I want. Right, so maybe as I get older, the antibodies might not be enough. So when I get given a booster, it just helps with the protection. Guys, when we talk about the COVID vaccine, some of them need boosters, right? The first one helps, but the second one just makes sure that we get that extra protection. Right, why is method B very good with dealing with an infection like tetanus? Guys, tetanus is a bacteria, right? And obviously that bacteria can have a huge bad effect. So what do we want? Why do we want method B? It's quick, right? It does what it needs to do. It does, all right, what it has to, which is to kill bacteria. Which is to kill the bacteria, right? And we want that to happen quickly, because with tetanus, if we get infected, right, infection could be deadly, right? So remember, if we're talking with bacteria, guys, bacteria we can deal with. It's living. We can get rid of it. A virus, we have to come up with a whole different kind of question. Okay, I know there's another question, but I'm afraid our time is running out. So guys, please make sure this is the end of microorganisms. Go over everything. Go over all the series that we have done. Start practicing past exam papers, right? And that is all for us for today. See you next time. Cheerio, bye.